So today we are carrying on the story of carbohydrates. We're going to have a look at how we can link structure and function. So this is a theme that runs throughout biology, and it's really important to get a grasp of it early on, as you can build it then as you go through. So just to be full and make sure everything's there, we're going to start with a bit of a recap of what we did last week and then pull that into polysaccharides in a bit more detail. So carbohydrates, we have our monosaccharides, we have our disaccharides, and we have our polysaccharides. Okay, so mono is one, di is two, poly is many. We're talking many, lots of. Okay, all of them linked together with glycosidic bonds. That's our bond for a carbohydrate. Okay. Right, then the other thing that we're going to sort of zoom in a bit and focus on for today is what is caused by this difference between alpha glucose and beta glucose. So let's just have a look at them again quickly. We had alpha glucose and we have beta glucose. So the chemical formula of both of these is exactly the same. It's the structural formula that's different. They're isomers. So alpha glucose CH2OH and beta glucose start exactly the same. Same ring structure, same stable molecule. Okay, the difference between the two is where our OH group is on that first carbon atom. Alpha is below the ring. Beta is above the ring. And then our new phrase from last week, beta behaves. So down, up, down, a beautiful zigzag. Whereas this one we go down, down, then up, then down. Okay, so there's our difference. And the impact this has is when we form a glycosidic bond. Okay, so let us move to another one so we've got plenty space. Okay, so keep those structures of alpha and beta in mind. Remember, we didn't draw in the H's because we're focusing on the OHs for today. Okay, so if we think about our glycosidic bond in a little bit more detail, okay, let's say we're working with alphas and we're working with betas. Our alpha, oopsie. Sorry, I just realized sometimes when I put my paper on my keyboard, it obstructs my mic. Okay, but you haven't complained yet, so hopefully that's all right. Right. So there's one of them, and here's the one next to it. Okay, so same story here. We're just looking at a piece of our rings. Okay, so when we have bonds like this sticking out into nowhere, that indicates there's something else that's bonding over there. So on our alphas, carbon 1 we are below, carbon 1, 2, 3, 4 we are below. On our betas, carbon 1 we are above, carbon 4 we are still below. And now we have a problem, an actual visible issue. And this visible issue is these guys are nicely lined up. Okay, we can easily take H2O removed and form our bond. Here, we've got a problem. Okay, we can't do this and have our OHs not lined up next to each other. Remember, an enzyme is going to hold these molecules together, but it's still catalyzing by holding them together, so they're still got to be close to each other. So what we've got to do is we've got to flip this second molecule. Okay, so this is what we talk about by rotate by 180 degrees for the second molecule. And what that's going to do is it's going to bring our OH up here. And now we can, ooh, yellow is a no good. Let's have blue for water. Now we're happy. So the question came up last week of which two do you circle? Do you circle OH on the left and H on the right, or do you circle OH on the right and H on the left? So I've always done it, OH on the left. That's the way my university textbook had it, so that's the way I followed. But I did a quick search and there's a whole lot of images out there with it done the other way around. So I think at the level at which you're working, it's not something that's going to be a problem, but the key point is that we're taking two H's and one O so that we can remove a water. Okie dokie. So we now have water being removed, but we've had an impact because remember that we have this CH2OH that sticks out. So CH2OH sticks out from ring. Now the thing is that what's going to happen with our alpha, all our CH2OHs are going to be sticking up in the same direction because we've kept our ring in the same order. Right, let's see if we can draw this. So there's our rings, the pointy bits are our glycosidic bonds, okay? So then all our CH2OHs are going to be sticking up. Okay, now if we think about it in terms of beta glucose, we have the same thing of a long list of rings. Ooh. But now what's happened, the first one was sticking up. But remember we've rotated the second ring. So we've taken the ring and instead of sticking him up, we've now rotated him. So he's sticking down. So the CH2OH is now going to be pointing below the ring. CH2OH. The next one, okay, we're going to have to rotate so that we can bond again. So he's going to stick up. So as we go down our polymer that's made with beta glucose, we're going to have one above the ring, next one below the ring, next one above, next one below. So we're sticking out in both directions. Now remember, think back a little bit further and consider hydrogen bonds from dipoles, e.g. OH. Every time we have an OH, we have that potential for a dipole. Well, not the potential for dipole, we do have a dipole every time we have an OH. We have the potential for hydrogen bonding. And that then starts to make a key difference here. Okay, so let us now have a little bit about think of how this is going to translate further into impacting our structure and our function. Okay, so let's have our polymers again. Okay, so this one has CH2OH, 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 whereas this one has CH2OH, CH2OH, CH2OH. So on the left, we have an alpha glucose polysaccharide, and on the right, we have a beta glucose polysaccharide. Now, think about it in terms of packing a suitcase. If you were wanting to pack your suitcase, would you want everything in a neat and nice and organized shape? Or would you want everything in a wiggledy-piggledy shape? And the thing is that if you get your wiggles and your piggles in the right direction, it helps. This is much easier to pack. Okay, it is more compact. Okay, this one is not so easy to pack. But what happens when you take two higgledy-piggledy things and put them together? They're a lot more stuck together than if they're smooth. Okay, so just from this basic, we're already making a difference in terms of what we're going to have in terms of function and how we link that to structure. So our alpha glucose polysaccharides make these nice compact structures. These are our storage. Whereas our beta glucose polysaccharide, much more structurally sound, much more hydrogen bonding, because we're bonding on both sides, okay? If you want to take two pieces of Velcro, they're going to stick together. They're not going to stick to the next piece, right? But if you have double-sided Velcro, can you imagine trying to even try and pull that apart? It's not going to happen because you're going to be stuck on both sides. So all these CH2OH is sticking out on both sides. We get a whole lot more stickiness going. And a whole lot more stickiness. Remember, hydrogen bonds are like Velcro. The more you have, the stronger it is. Okay, don't go talking about Velcro when you're writing answers for exams. Okay, this is just an understanding type of description. Okay, so structural, we have just the one we're going to deal with. Cellulose. Storage, we've got 3. Okay, so amylose, amylopectin, and glycogen. Okay, these two form starch. But it's just a word to the wise to actually think of them in terms of, first think of amylose and amylopectin, then think of starch, rather than thinking of starch first and then its components. Because you're working at the level now where you need to be familiar with the components, not focused on the higher level things. So focus in on the components first. Think of it as plant store, amylose and amylopectin. Right. So these are all compact storage molecules. And this is increased connectivity and hydrogen bonding for strength. Right, so that's them. Let's get to know them in a little bit more detail. So we're going to split now. Between our storage and our structural. Okay, so we have, how do I want to write this because my paper was not the same dimensions as this video is. Okay, so let's go like this. We have amylose, we have amylopectin, and we have glycogen. Okay, so these two are what forms our starch grains. This one forms our glycogen granules. So we have then a difference. This one is for plants. And this one is for animals. It's a fish, I promise, representing the animals. Okay, so all organisms, our multicellular organisms, are going to need this storage system because we need to consider the fact that glucose is really reactive. Glucose is a mess. Okay, it's reactive. It affects osmotic potential, and if you affect osmotic potential, then you've got that whole risk of cell rupture and things. If you're an animal cell, for a plant cell, you're getting problems with turgor pressure. So we don't want glucose around. Okay, we need to have it available so that when we need it for respiration, then it's there. But we don't want it sitting in the cell as it is. So just bear in mind that everything you've done at IG in terms of respiration, equations for respiration, you are expected to know that as you move into AS. For AS biology, we don't touch respiration in detail. It fell under the split for A-level biology, but all of that background knowledge from IG, it's assumed you know it. So be comfortable. You need to go back and revise it. So thinking now, diverting a little bit to structure, amylose, let's actually split here. So amylose is helical, okay, whereas amylopectin is short branches. Now, we also talked last week about how we can label the type of glycosidic bond by using the carbon numbers. So whenever we're making that chain of glucose molecules, it is 1,4 bonding. As we just said, it was the OH on the 4 and the OH on the 1, and they line up together and make a bond. So this is 1,4 glycosidic bonds. Okay, so amylopectin also has to have that same chains before we can do anything to it. 1,4 glycosidic bonds. But now we've said amylopectin is branched. It's not just one long line. It has branches coming off. And the only way to get those branches coming off is to use that carbon that's hiding there in the CH2OH. Okay, so he's carbon 6, and he starts a new chain. Now, if a chain is 1,4, we're bonding to a 6, we need a 1. So we have 1,6 glycosidic bonds. And this is to form branches. Okay, so if we think about it for glycogen, oops, similar sort of thing, okay, we have extensive branches here. So extensively branched. Okay, but again, we've got to have that same structure of the links. To get the links, we need 1,4 glycosidic bonds. To get our branches, now again, we're going to bond a 1 to something. We've got to stick it onto that chain. The one that's sticking out, ready to bond, is the 6. So 1 to 6 glycosidic bonds. And these form the branches. So, why have we separated it between plant and animal? Okay, well, they have different things. They have different requirements. Okay, amylose and amylopectin forming starch, making our reserve and our store for our plants. Glycogen being our store for our animals. Okay, why do they need different things? It's because they have different energy requirements. And this leads to different storage molecules. If there was no difference in function, there would be no difference in structure because the two are so tightly linked. Okay, so the fact that glycogen has a different structure to amylose and amylopectin means that there must be some reason. There must be a functional difference here. And it's quite simple in that the demand for increased ATP production is higher and faster in animals than it is in plants. Okay, so plants are still enzyme control for everything. They still have active transport. They still need energy for things, okay? But they have less movement and less rapidity of needed of release of energy. Whereas animals, you've got movement, you've got thought, you've got all these other things going on, and that has a higher energy demand. Now remember that glucose is all locked up in these storage molecules. So in order for respiration to occur, you've got to have an enzyme reaction to release the glucose. Glucose got to get to a mitochondria. Inside the mitochondria, now the glucose is going to go through respiration, and we're going to produce ATP. So with a whole series of events, the more glucose you can release at once, the faster it's all going to happen. The faster we're going to get respiration, the faster we're going to get ATP production. And then we can use that ATP. So when we have more branches, we have what we call more terminal ends. Okay, a terminal end is a final end. Okay, and that is where our enzyme locks on. Our enzyme doesn't go onto some random spot in any of these molecules. Okay, we're always using the terminal ends to put on and to take off. And the more terminal ends there are, the more quickly we can put on and the more quickly we can take off. So we keep talking about taking off, but now think about it. You go and you eat a big carbohydrate-full meal and your blood sugar goes up and the glucose goes rushing into your cells and you don't have cell walls. So the thing is that just as quickly as you want to be able to take it off for respiration, you want to be just as quickly able to put it back into storage when your glucose goes up. We don't want that glucose sitting around in an animal cell for any longer than it absolutely has to be. So again, more terminal ends means more rapid storage of glucose. We don't have glucose molecules sitting in a queue waiting to get stored. Because if they're sitting in a queue, then we have a problem. Glucose molecules sitting in a queue don't behave very nicely. They're reactive, they're soluble, they're going to cause nonsense. So they come in, they get stored, finished, end of story. Okay, more terminal ends make that all possible. Right, so if we now have a look instead at cellulose, we have just the one, okay? Remember that there are other structural polysaccharides out there. It's just that cellulose is the only one that we work with. We do have to restrict our knowledge. You can't learn everything in 1 year and forget about learning anything for the rest of our lives for biology. We need that potential to take it further. Okay, so cellulose, he is found in, you should be very familiar with this, your plants. Okay, so we've said that he's got a whole lot of increased connectivity so that we can increase the hydrogen bonding between molecules. Okay, so we are doing glycosidic bonds. So we're making chains. Okay, with, whoopsie, didn't do it like that, 1,4 glycosidic bonds. Because we're still just flipping. It's a rotation, okay? It's not changing the carbon numbers that are involved. Okay, so chains of 1,4 glycosidic bonds. Okay, and we like to write it as well as a 180-degree rotation between consecutive monomers. Okay, sounds slightly fancy, but you're at the level where you should be familiar and happy with words like this. So monomers, remember, is one unit of our polymer, consecutive, next to each other. So we're not going to say between neighboring single rings. Okay, that's far too basic, and it's far too many words. We want our our writing to be concise when we're answering questions. So 180-degree rotation between consecutive monomers covers the whole thing. Right, so then we also have extensive hydrogen bonding between chains. And this gives us high tensile strength. So let us think about how we build these things up, okay? So let's say we have one chain, one molecule, one polysaccharide. We're going to take 60 to 70 of those. So 60 to 70 cellulose molecules. We're going to bundle them together, okay? Hydrogen bonding, stick them together, and this forms a microfibril. Okay, now we are talking 10 nanometers in diameter. These things are tiny, okay, but they're strong. We're actually now going to take a whole lot of microfibrils. We're going to put hydrogen bonding between them, and we're going to make a cellulose fiber. Okay, so this is about 50 nanometers diameter. So this all is more and more hydrogen bonds. And when you have more and more hydrogen bonds, you have more and more strength. And that then gives us that strength of that cell wall that can withstand the osmotic pressure, that we can deal with this whole turbo pressure, and we can have whole herbaceous plants standing upright just because of these cell walls and the interaction with pressure. So, crucial. Okay, structure links to function. If we have a different function, we have a different structure. If we have a different structure, we have a different function. So get familiar with it at carbohydrates and then run it as a theme all the way through. Whenever you learn a function, why has it got this function? How does it link to the structure? Whenever you learn a structure, why does it have this structure? What's the function that I'm linking to? Okay, again, we're helping to consolidate, to bridge, to link, and to make everything a whole lot more solid.